I'll condense it to where kind of meat and potatoes, if you will. And then on a Thursday, we'll also condense chapter 12 and 13 to hopefully get it into a single presentation. It's not as much technicality in chapters 12 and 13 as far as just observational type of information. So I was thinking we had an extra week and we don't. So I think next week's the last week of what I have scheduled for lecture. And looking at our schedule, the last week of classes I actually scheduled for a test and then a review to get ready for the finals. So a week and a half, we're going to take our 4th unit exam for the semester on Tuesday the 6th, I think, whatever date that is. It's coming up pretty quickly. We'll take our 4th unit exam covering chapter 10. What do you think you might be doing on chapter 10 test? Yes. There you go. So we certainly think we're going to take 3 days on it. So
it's just chapter 10?
No, but for the chapter 2 component of unit 4 exam, we'll talk about evolution and then evidence of evolution. Okay. And then we'll do the final, very final thing. So let's get back into that. Realize I was recording already. Okay, so we are going to go back and be concise with interpreting the dihybrid cross. When I created the file, I was apparently asleep or close to asleep, so we'll go back and rework that.
We're doing more examples.
Yeah, we're going to go back with the example of the tongue rolling freckle example, but state the known genotype of the parents, so we can better understand the dihybrid cross, and then as well as review that little rabbit scenario handout that I gave you guys. All right, so, and if you, of course, obviously, just like any other day, if you have questions, ask those, and I'll hopefully be able to address those for you. So just to review where we left off, we were doing a dihybrid cross, and we were going to analyze 2 traits and whether or not they would be potentially passed on to offspring in this potential mating. And so we were looking at tongue rolling. We've identified that the ability to roll one's tongue is a dominant trait, as well as freckles are also dominant traits. We're going to those. So what we want to do, and this is the important bit that I kind of left out, is let's cross a female that is heterozygous for both traits. So how would we write her genotype? Okay, so big R, little r. Okay.
Oh, there's 4.
And big F, little f. So, or I could say heterozygous for both traits. We're analyzing both of those. And then we're going to cross her with a man who is homozygous recessive for trait 1, which is our tongue rolling. So how would you write that genotype? 2 lowercase r's. Yet heterozygous for trait 2 is freckles, which is big F. Each one of these alleles. What we need to first do is figure out what the potential allele combinations would be in each the man and the woman in their eggs, the female, or the sperm for the male. And so if you have your handout for page 6, the first thing it says determine the combination of the alleles in the gametes. Now you want to underline, circle the word gametes. So you know the type of ploidy the gametes have is what? Haploid for each trait. So even though there will be 2 letters in these combinations, the letters will be what? There'll be one of each. Okay, there'll be 1 F and 1 R. And whether they're dominant, recessive, that's what we're trying to figure out. But they'll be haploids, so they won't have each of the traits. And so we're trying to figure out what's the combination between the freckle allele and the tongue rolling ability allele. What would be their combination? So on the female, we're looking at trait 1. We said she's heterozygous for the tongue rolling ability, so one dominant and one recessive. We can move each of these alleles across. R over here, and also bring it over here for the dominant. And then I could do the same thing for the recessive allele. I'll bring the recessive allele over. And then to complete the table, I bring down the dominant trait. In this case, pretty good handwriting skills too. Mouse. Anyway, you can kind of hopefully get the gist. And then the second combination. Does this make sense? Okay, any questions? Yes. The order does not matter. I would put it. It doesn't matter, but I would put it where your brain can handle it, and I would be consistent. I would always put maybe trait 1 first and trait 2 second. For the gametes, I would just put trait 1. When we look at the offspring, I would actually put the dominant allele ahead, just so you can visually think recessive or dominant, whether or not it's present. But it doesn't matter. But the more you jumble it up, the more your brain's going to take it. So I would keep it simple. But in technicality, it doesn't matter. So go ahead and complete your allele combination for the female oocytes and the male ova and the male sperm. You have one copy of each allele.
On our test, are the people going to be labeled on test with trait 2 or 1, trait 2 or 2?
I should, yeah. Make it consistent. The male is homozygous recessive for the unborn. When you go through, you can check your work really quickly. If we look at the combinations between a female that is heterozygous for both traits and a male that is heterozygous for the freckles, but homozygous recessive for the tongue rolling ability, we see a nice little mixture of combinations here of the alleles. So we have 2 dominant alleles. So those, both are a double check. How many variations does the female have? Of the 4, how many are different? 4. And of the male, there's only 2. And so when we get to the dihybrid square, you notice that the chart gets kind of extensive. When 2 of the 4 or all of the 4 are identical, you really don't need to repeat it 4 times. Maybe just do, instead of copying, writing the exact same thing, right, you can kind of just deduce the percentages. So I'll address that, point that out in just a second, what I mean by that. Is everybody okay on how I created the, or determined, and create them, determine the allelic combination between these 2 parents? For now. One thing that should stand out to you is these are haploids, because I only have how many copies at one time? 1 copy of each allele. That tells me this is what type of cell? A haploid cell, which is a gamete, sex cell, sperm cell, egg cell. We have to know these combinations so that when we cross them for fertilization to form a diploid offspring. So we always have to do that first. We can go through and we can go through and I'm going to back up this. But if you look at your table, we can then go ahead and predict the combination of those potential sperm and potential eggs, look at the probability of what characters an offspring might have. And so you notice the chart gets a lot more extensive. It's now 4 by 4 because each woman could produce, in theory, 4 potential oocytes. The male could produce 4 potential sperm combinations. So all you're doing is taking those egg allele combinations and then moving them over here where the arrow is, 1, 2, you're just physically just placing in this, what I have here in the shaded area, you're just physically taking the information from the egg table and just placing it over here so we can read it. And then you would do the same thing for where it's labeled male. In this case, I have a male across the top. It doesn't, as long as you're just consistent in how you're placing it. So the male sperm allele combination, it would just transfer those 1, 2, 3, 4 over across. Does that help? It's like the homework, but just everything else. All we're doing is just moving the potential egg combinations into one column and the potential sperm allele combination. Yes. And now what you're going to do, instead of just one trait, you're going to cross 2 traits. So each table that I have shaded white, or lacking any shade, would have how many alleles? 4 alleles, 2 of each type. Okay, so and this is what will determine if the allele combinations are homozygous or heterozygous, or whatever the trait may be.
Mix them up while they're here?
You can, yes. What I would do is keep the alleles together, the letter F's together and the letter R's together, and I would also visually put the dominance of those 2 over on the left-hand side. When you start adding multiple traits, sometimes it's useful to keep different colors, like I tried to show up here in the presentation. You notice it's easy, especially when your handwriting, if you're writing all caps or whatever, it kind of accidentally mess up. I'm sorry? So a question that I've seen just twice in just a few minutes that we've been looking at this is, well, how do I interpret this? You need to interpret each allele combination separately. So you don't interpret all of the alleles, you don't interpret all four together. You interpret one gene, whether it's homozygous dominant or recessive or heterozygous. And then you do the same thing for the same trait. So just like I introduced the female, I said she is heterozygous for both traits. But for the male, I said he's heterozygous for one trait, yet homozygous recessive for the other. So you're going to apply it to both. You're going to study each gene individually in your interpretation. When you do the cross, you should end up getting something like this, and you'll notice that I didn't fill in all four columns for the male because you said really how many variations you have. He has two. So I could have just as well duplicated this over. But you notice that I filled in all the rows for the female because she had how many allele combinations? She had 4 allele combinations. So the rows that are, the columns that are missing would obviously be duplicate to the column prior to it. And so if I go through and put these combinations together, in the first column, I can see the genotype. How would I call the, for the freckles? In this first column, the genotype is what? Homozygous dominant for freckles, yet for tongue rolling, they are heterozygous. And this also addresses the question, does it matter the order in which you place them? No. Okay. I suggested for you to actually put the dominant one first. Okay? So that I know which phenotype is going to be expressed. If we go through, moving down to the next combination, for each allele, the next combination, the combination, the offspring would be heterozygous for each trait. The dominant and recessive for each trait. Moving to the third row, the first column, we would say this combination is now for freckles, homozygous dominant, for freckles, but for tongue rolling they are recessive. And then the last column, excuse me, first column, last row, heterozygous for freckles and also recessive for the velvet roller type. Looking at the additional combinations, moving down to the third row, we can still, right, in this next combination, the potential offspring could be what for each trait? Heterozygous for each trait. Looking at the second row, we could say they are homozygous, recessive for freckles, but heterozygous for tongue rolling. So just kind of falling together a little bit how I would interpret those. Any questions on how we came up with those genotypes? You might want to label on the top of your handouts and identify that these are in the potential offspring. And we know this because what type of ploidy are we seeing here? They're diploid in the cross. We use haploid cells to create diploid. I know that they're diploid because I see not just one, but I see 2 copies of each allele. So this, if the fertilization happens, that baby would have the right amount of genes, alleles of each baby. So I'm going to make a note that we know this is an offspring because the cells are diploid for each trait. Now we should go through and interpret the phenotype for each of these traits. So let's look at the phenotype. You're going to have to identify each trait. The tongue rolling, or freckles. Easy legend. So look at the phenotype and just kind of squeeze it in there in the bottom. Go ahead. Easier to interpret in the square. In the handout that I gave you guys. So just write in there the phenotype for each of those traits. The genotype. So the phenotype is what? Freckles or tongue roll. We're just looking at what physical traits. Expressed, yes. As long as there is at least one dominant allele for that particular trait should be expressed. starting to click together for you guys. So if we go through and actually look at the phenotype, right, based on the genotype, we can look at some combinations. And so just check your work. Anytime you see a dominant for each trait, that trait should be expressed. So anytime I see a capital F, I should say that that person, that potential offspring would have the expression of freckles. Anytime I see an uppercase R or a dominant allele, I should see those potential people have the ability to roll their tongue. And so we're just going to go through and figure out what's the mixture of those combinations. Right? So you can go through and check your work. And there was a question?
Yeah, so the letters represent the genotype, and the phenotype is what they are.
Yes.
What they represent.
Yes.
Especially the phenotype.
Well, what makes it complicated is we set our legend up improperly. It's when we get messed up. That's why I'm really articulating in the third day now, set your legend up. Additional questions on how the genotype can dictate the phenotype, or even what those terms mean? I'm looking at the phenotype of someone. That someone should be a diploid cell-containing person, so we have each allele expressed in there. Another question that we had is, well, then how do we determine the proportion of a particular trait? Like, what percentage of those? There's lots of different ways that you could go through and express this. I could say, well, what proportion of these potential offspring cannot roll their tongue? So let's go through, let's count them up. 1, 2, 3, 4. 4 out of 8 cannot roll their tongue. So how many cannot roll their tongue? 50% or half. And then we can extrapolate then how many could? 50%. You can only look at yes or no case. What about, those who have freckles? What percentage of the offspring have no freckles? How many have no freckles? 1, 2. 2 out of 8, so that's how much? How much? 25%, a quarter of them. Yeah, 1/4. Okay, so 25% do not have freckles, so we can extrapolate them. 75% would have freckles. We can even get more complex about combining the alleles. How many have no freckles and cannot roll their tongue? 1 of 8, so 1/8, half of a fourth, 12.5%. So
Are you going to expect us to do that?
no, I'll keep it. I thought maybe simple, 25%. But I'll probably do it in 25 quarters. 25, 50, 0, 100.
I don't want to do that.
It's a little crazy.
It is. It's pretty wild.
That's part of the news.
It's very difficult. Hopefully it's falling together for you a little bit. You can certainly see the complexity of doing a dihybrid compared to the monohybrid cross. And I gotta let you know the Punnett star is not the only way that you can do this. I mean, there's more simple ways to do this. There's more complex ways to do these guesstimates. But I like this that it just stares you through and hopefully draws out the genotype, how it relates to the phenotype. And then when we see it in quadrants like that, we can begin to put together proportions and percentages. It's just a predictive tool. It's not a guarantee that if these two people had 16 kids, one of the 16 would not have pregnancy. What's the family? What's the family in Arkansas? Of course they're in Arkansas, right? Duggar. So what is the Duggars?
Yeah.
Right?
Yeah.
The Duggars. So of their 25 kids, they all look a little bit different. They're not all exactly. So it's a probability predictor, not a guarantee. There's no guarantee that a kid is going to. That's what she said. Alright, so we went ahead and interpreted the phenotypes using our little key from earlier. That is a dihybrid cross. Any questions on that before we kind of put some additional genetic application to that tool?
On the worksheet, it asks what kind of gametes would be produced by the spotted brown rabbit or the black rabbit. I don't know what that means.
That's what you missed this morning.
Oh, okay.
So, all it is just mixing the allele traits. So we looked at the allele combination
Okay.
based on their phenotype. Now remember, looking at the phenotype, let's look at the little rabbit thing real quick. Okay? Let's look at the rabbit scenario. Okay, we say we have a female spotted brown rabbit whose mother was solid black. So we're having to decipher her genotype based on known dominant recessive traits. We now know what her parents are. We know about her traits. Okay? And so this person should have what type, or this rabbit should have what type of ploidy in their cells? In their body cells, they should be diploid. And so how many letters for each allele should you have? 4. For each allele, you should have 2. We're looking at 2 traits. That would be 4. So how many S's should you have? 2. How many D's should you have? 2, or whatever letter you want. Yeah, you can pick whatever letter you want. Usually, most of the S and U. Yeah, that's fine. Yep. But the gametes would be haploid. Okay. So what is, right, so this rabbit, I'm trying to read upside down, this female spotted brown, she's spotted. Spotted is dominant to solid, so she could either be homozygous dominant or what? Heterozygous. We've got to research more. Her mother was solid, so she must be, solid is recessive, so it tells us she has at least one recessive allele, so she would be considered what for her? Heterozygous. So large S, lowercase s. If you just
I think the legend is what the hardest part. It's not reading now and figuring it out. It's like the most difficult part.
So let's just keep going. So we know that the female is heterozygous for pattern because she's spotted, and her mother was solid. So it tells us that she has at least one recessive allele, which tells us she's a heterozygous for the color, or pattern, excuse me. She was brown though. Brown is what? Recessive, so 2 lowercase allele. Brown is recessive, so she's expressing 2 lowercase. So how would you write her genotype? Very good. If you use the letter S for solid or spotted, she'd be heterozygous.
But you don't need to know her whole genotype to figure out what her gametes are.
I do need to know her genotype.
But it doesn't matter what her mom was, as long as I know that she's whatever.
I wouldn't be able to know that she has that recessive allele for the spotted ability if I didn't know one of her parents.
Yeah, but her gametes are all going to be the dominant, right?
Her gametes.
Gametes are haploid.
Yes, and so half of her allele gametes will get the dominant, and half of them will get the recessive. We wouldn't know about that appearance of the recessive trait if we didn't know her parental heritage.
Okay.
So we do need to know that. If she had only recessive traits, then no, it's not. But since she has a dominant trait, we don't know if dominant traits are expressed because the organism is heterozygous or homozygous dominant. So it is important. So let's look at that. What would her allele potential, right? So looking at her eggs for trait 1 is using the letter S. Half of her eggs would have which allele? Dominant. So we're write a big capital S here. And then half of them would have small recessive. But we're doing a dihybrid cross, so we need to figure out what our other allele combination is. So we said she is what for color? Homozygous recessive for trait 2. Allele 1 and 2 are both lowercase d. Well, that's probably just right. She's homozygous recessive for both, so you need lowercase d's for all. years repeatedly in its call. I would not, I mean, you could, I mean, like I just said, 50% are dominant for one trait, recessive for the other, 50% are recessive for both traits. But we're not looking at the phenotype yet. Yes. The male, what trait did he have? He's solid black. So solid is recessive, so two lowercase s's, or you could say homozygous recessive coat pattern. Two lowercase s's, or whatever letter you use, two lowercase for that pattern. Yeah. And in a body cell, how many copies of each allele should there be? Two. So on the very top, 1A, 1B, the genotype of the animals, we should have four letters. Thank you. Let's leave you right there. You need 4 letters. 2 S's, 2 B's. Because a reproducing, mature organism in their body cell, because they got half their alleles from mother, half from father. Alright. So now let's look at the male. Let me revisit him. He's black. Is he solid black?
Solid black.
He's solid black. And so we said he is homozygous recessive, so 2 lowercase s's. And he's a black coat color. And his parents are also homozygous, so he's homozygous black. So he has only one allele combination for his potential sperm cell. They are all recessive for the coat pattern, so it's solid, lowercase s. And he's going to carry only the dominant allele for color, large B. So both of these are going to be B. Yeah, so double those out.
Yeah, makes sense now.
Now, if you notice the titles, it says female eggs, male sperm, and you analyze the crosses, each allele is only in there once, because those will potentially fuse. We took from those quadrants in there, those potential gamete combinations, and we're moving them over to the backside to kind of mix them up. Okay, so you're going to move those pairs together, just like we did for the tongue rolling scenario. Yeah. Technically,
I have to erase it.
can bring a recessive allele to the party, like a female would, so. Their babies could have, what kind of babies though? Or, their babies, their grandbabies, their grand rabbits, could eventually have brown babies, because the recessive allele is in there. If we went ahead and did this again, we would have 2 generations. But you could. So, what percentage of, in this mating, of the rabbits are brown?
None of them.
Brown are 0. What percentage are black? 100%. What about spotted?
Most of them.
4, 8.
4 to 8.
4, 2,
1 half.
4, 1
Oh my God.
Nice deduction.
You are on it today. I hope you don't have an algebra test today.
Already done.
I can tell.
The skeptic of me. Alright, so percentage-wise will be spotted, you said, 50%, and solid, 50%. You could do that anyway. What you can do is on print page 5 and 6 from your handout and just make up one and just practice. I'd like to go ahead and move on so we go ahead and conclude chapter 10, which is not actually complete 2 weeks experience. Close. And talk about what else and what this means. So, if you weren't able to print chapter 7 handout, I'll do it for yesterday afternoon. But we're going to look at some of the questions that you guys brought up about why is this? And one of they call it now a law of genetics or inheritance is called Mendel's law of independent assortment. And this just restates what we kind of alluded to when we looked at meiosis. And what it tells us is that chromosomes will sort independent of one another during meiosis. Chromosomes will sort independent of one another during the process of meiosis in a parent. Something that is about to reproduce. So our chromosomes will sort independent of one another. In other words, like for example, in the human, how many chromosomal pairs do we have? 23 chromosomal pairs. But it tells us is chromosome 1, when it duplicates, chromosome 1 won't always sort with chromosome N, which won't always sort with chromosome whatever. Okay? They sort independent of one another.
This is kind of like random
It is being,
Is that what that means?
it's contributing to it. Okay? What this most specifically means is our chromosomes aren't paired up together. So chromosome 1 doesn't have to adhere to chromosome 2, doesn't have to adhere to chromosome 5, whatever. Chromosome 1 does its own thing, chromosome 2 does its own, all through the 23 in the human, they're going to be moved separately. What that means is all the genes for chromosome 1 are not adhered to all the genes for chromosome 5, that are not adhered to all the whatever. Okay? So you think about, for example, there's chromosome, is it not 15 that we're looking at for color, something like that, right, in human, that chromosome would move different than chromosome 5 where ATP synthase. Okay, so you don't find those two pairs being moved together during the meiotic push-pull process. That's all that's saying. is they're separate when they get ready for division. Okay? And here's another way to look at it. One gene on a particular chromosome does not express the allele expression of a different gene on a different chromosome. Bless you. Okay? So just using the examples that I gave, the gene allele frequency combination for ATP synthase has, because it's on chromosome 5, has no control over the expressions of the allele on chromosome 15 for your hair and eye color combinations. They're completely separate. They move separately. They don't directly influence one another. It's almost like separate people. Okay, well, I'm shuffling in here. We're separate people. We would be separate chromosomes. If I were trying to divide us up, I wouldn't always have you pair together like in your table or something like that. You'd be separate. You could move about however you want. Does that help a little bit? And so it's dealing specifically with the chromosomes aren't grouped together when they move. They're just a chromosome, single little unit. Then. Yes, usually. There's always exceptions. But for Mendel's work, that's the case. That's why he's looking at that pea plant. We said it's either purple or white flowers, or it's just. I mean, it was very straightforward. Those chromosomes are moving separate of one another. They're not having influence upon one another. And so the question is, well, you're thinking why, but what are some exceptions? Some traits, and you guys already identified this, we said some traits aren't simple like these Mendelian traits that we've been studying. Some are, which is good, it gives us diversity. Some traits are actually controlled by multiple genes. These are called what you call polygenic traits. Polygenic traits are those traits that are controlled by, you look at the word, many genes, multiple genes. And in these cases, in those polygenic traits, you're not going to have, like Mendel saw, either tall, either short, either purple, either white, either green. There's none of that oversimplified business that we kind of see in the pea plants. And there's a lot of variation. The example we gave in class the other day was like skin pigment. Okay? We're not all just albino or pure black. There's a gradient of colors and just the human color pattern expression, for example. And so there's a variation, not just one or the other. And so typically you'll find like an average expression for a lot of genes. You'll tend to, within that grade, there tends to be an average type of expression for that particular allele. The examples that your book gives of this, we can see, for example, like our height. We're not all either 5 foot or 7 foot, 4 foot, right? Whatever. There's a range. And we can think, in America, the average height of a male is whatever, okay? Whatever. Okay, so you can find on these polygenic traits, there tends to be kind of a bell curve type of association with them. It's not one or the other. You're not either 4 foot or 7 foot. You're a range in there, for an example of height. So I gave that example. Another example is like milk production. You think about dairy cows. Dairy cows don't always produce, say, 5 gallons of milk or just 20 gallons of milk. Right? There's like a range and there's an average based on their gene expression rate. So there would be a cow, person, goat, whatever. Weight. We're not all either 50 pounds or 300 pounds. And you know that the weight is not just only genetically controlled, but our genetics certainly have a role in it. Affected by other things outside of what fit your body. And we gave an example of like skin pigmentation. Or you could look at color patterns of anything that has color, not just humans. What kind of traits are these called? genes can influence some phenotypical traits that we see expressed. Is that a little bit more comfortable? Not as scary as you say usually. Bell curve, as I mentioned, is kind of what you'll see for a distribution for our polygenic traits. The good example in your textbook does show this gradient of, for example, skin pigmentation. Not either one extreme or the other, just a combination of expressions. You want to do a cross on this? Colin! What percent is that?
I don't know.
Alright. And the usually is, some traits may actually have a third phenotype instead of just A or B, there actually may be a third phenotype. And in those types of phenotypes, we call that, you may have the expression that's called incomplete dominance. So there's not full dominance of a trait. You might actually have two dominant phenotypes expressed at the same time. And it's called incomplete dominance. You may actually see two dominant phenotypes expressed at the same time, and they may actually, depending on what the trait is, it may actually be like a blending of the characters. So if you have maybe a heterozygous type of, I think it is. I'm not sure about the hazel. I think it is. I'm not sure. I'm pretty sure it is, but I'm not getting like no with definitive things on that. So you get kind of a blend of the traits. Some examples that you guys are looking at. I'm not sure about the eye color, but if you look at brown rabbit fur, actually a combination of the red and white allele. Gray sheep, you ever seen a gray sheep? Blend of the black and white. You ever heard of sickle cell anemia? That's a blend of heterozygous people who have both a dominant allele for normal hemoglobin assembly and a recessive allele for improper folding of the hemoglobin. So some of their hemoglobins are good and some of them are not good. It causes the whole cell to change completely. Okay. Wavy hair. A combination of parents who have curly hair and straight hair, so it's a blend. It's not full curly, not full straight. When I described incomplete dominance, I think I actually described codominance. So let me restate, double check what you wrote on incomplete dominance. The proper way to describe incomplete dominance is in the blending, like I mentioned, instead of when I misspoke when I said two dominant alleles. If you wrote down two dominant alleles, did you write that down? Strike that or circle and put it down for dominant instead of that. So in incomplete dominance, correct. It's a blend of it's a blend of phenotypes. So in heterozygous organisms, they don't fully express the dominant trait. They express both the dominant and the recessive trait. Very okay on that? Yes. In incomplete dominance, incomplete. There's a blending of both the dominant and recessive phenotype. And the examples are red. One of the other parents was white, so then homozygous white. This one's a heterozygous, so the third phenotype is brown, not white or brown. I mean, white or red is third phenotype, the blue of the brown. So, okay on incomplete? It's just a blending of the dominant characters. Where I was heading and getting ahead of myself is the role of codominance, where you actually find a couple of alleles expressed, and then two alleles are working together. One does not dominate over the other. Hence the term codominance. A more prominent example of codominance is in human blood type. Do you know the types of blood that we have? A, B, and O. Okay, so in that case we have you can even have A and B. Do what? I don't know about the rarity, but it'll be the most
There's only 4% population.
AB? Okay.
I looked it up.
Okay. So it is the most rare? And what's the most common?
I think A and B are both. But not A and B.
Does anybody know what the A and B represents? What the A and the B, we can use those to translate to alleles. They actually represent proteins in the membranes of your red blood cells. So if you remember, yes, it's a glycoprotein, a sugar protein. So if you remember way back in the beginning of the semester, we said cells have membranes that have proteins in them. And our red blood cells have specialized glycoproteins that we use to type our blood, which has to do with our immune response, basically. And so if you have the A allele, you would have blood type A blood. Or if you have the B allele, you would have type B blood. If you have the O allele, you don't have those glycoproteins expressed at all. If you have AB, you have both of those alleles. So an example of codominance, instead of just A or B, I could actually have AB like their father, 4% of the population. So it could actually have expressed the codominance itself. That's found on chromosome number 9. We already identified the different types of alleles. You have A, B. They use the letter O, 0 is inactive. And really there's 3 allele combinations, A or B or none of them being expressed. And you can go through and mix them up, what you guys kind of went through. And so if you're getting one allele from each parent, someone could be A, they could be AB, or they could be A, O, or both A. Someone who has a allele combination B, they could be AB, BB, or yeah. Or someone could be, we already identified them, or they could just be the positive and negative is dealing with a completely different glycoprotein on the membrane, and it's called the it's called the RH factor in the RH chain. The rhesus monkey. Okay, so they abbreviate it RH. It's identified in the rhesus monkey. And so it's a different protein. If you're RH positive, you have that protein. If you're RH negative, you don't have that protein expressed. So then you could add in there a second protein in there. And so we could do a dihybrid cross. You could do a dihybrid cross with the blood type and the rhesus or the RH factor. So these are different combinations. Alright, so we went through and looked at the different combinations. So just 3 alleles, I mean really we could get 7 different combinations. Common question, excuse me, 6. It's a combination. Question is, we apply this in donating blood or receiving blood. Okay. Who could donate blood to anybody? Type O, because they don't have the protein. So if you put that blood in someone else's body who's A or B, they don't have the ability to, there's nothing to recognize to attack. However, who cannot receive any could, right, type O people can give blood to anybody. Can they receive blood from anybody? Because anybody else can have an A allele or a B allele, which their body would attack. They can only give O and only receive O. Somebody has blood type A, they could receive type A blood or type O blood. So this is an example of what's called codominance. And the big deal is the A being, you can see them dominating, being expressed at the same time. Codominance. Does that make a little bit of sense?
And depending on the negative or positive, you also have to look at that too. Like if you see the positive one or the negative.
You shouldn't. You can if it's the first time.
Yeah, that's what I heard. But I
If you receive it, if you're say if you're negative, Rh negative, and receive Rh positive blood, one time you can receive it, your body will try to attack it and it'll build antibodies. The next time you receive it, your body says no. And so the big deal is with pregnancy.
Because the first time you brought in the second time,
It's harmful. So the concern is when women are pregnant, they're growing a completely different person with hopefully a completely different genotype in them. And if the father is Rh positive and she's Rh negative, the baby might be Rh positive. And so her immune system will try to attack the growing baby. So that's the primary deal. It's an adult.
so is that maybe because of having a miscarriages?
Could be a cause. There's many, numerous
Obviously, it's
reasons.
not one reason, but
No, it's certainly one of many reasons why. Codominance. Okay. All right, in your book, figure 1015 illustrates for you. Right here, you can see our little codominance, both A and B expressed. And you as a person out there in the world, you know that environment can also influence gene expression. For example, your diet can have a huge impact on your genetics. It's huge. So you hear about eating right, not just for having like what you think about for a healthy weight, but it does can have a positive or negative influence on the health of your cells at the genetic level. Smoking, the toxins can certainly negatively influence gene expression. For example, leading to cancer, right? Lung cancer or mouth cancer, whatever. Changing that cell cycle, mutating the DNA. Soil pH, good example, right, from your textbook, right? Hydrangeas, really sensitive to the pH of the environment. So even though genetically speaking, these hydrangeas, depending on the environmental condition, it'll change their genotype or phenotype expression. Temperature can have a huge impact on certain proteins and genes and their expression. A cool example outlined in your textbook are the Siamese cats. They got that cool coat pattern, and it's just because at the ends of the body, the temperature is a little bit cooler, and so their proteins express differently than where the body is warmer. That's just a temperature effect on protein expression. So we can keep going on with the environmental influences that affect gene expression. That's going to conclude our discussion for chapter 10. I want to take the next 5 minutes and just go ahead and set up chapter 15, or chapter 11, sorry.
That's why I jumped.
Chapter 11. Chapter 11 is dealing with, do I know? Evolution. Is it a dirty word? No. What I want to do, this is chapter 11. I just want you to set up, kind of get you ready prime for reading chapter 11. So, evolution is a scientific what? It's not a law.
Theory.
It's a theory. So, what do we know about theories?
They haven't been proven.
Haven't been proven without a doubt, but what?
Plausible.
They're very well accepted. They're very plausible because there's a whole heck of a lot of what? Evidence out there, okay, to support this concept of evolution. So before we look at what evolution is, I want to identify what evolution is not. Okay? So I think a lot of times we run with what evolution is not and then think of it as the truth. And so I want to dispel those. Primary thing is evolution has absolutely nothing to do with what? Religion or belief. Okay? It has nothing to do with religion. There's no religious, from the scientific community, there's no religious implication in it. Okay? We identify it as a scientific theory, which tells us there's a whole heck of a lot of evidence out there to support it, but it's not a law. We can't quantitate it. We can't put a mathematical application to it. It's going to take a whole lot of work to actually dispel a scientific theory. I mean, it could be, but it's going to take a lot of new evidence out there to dispel that. We identified nothing to do with religion or religious belief, so you can be as Catholic as a Pope and still believe in the scientific theory of evolution, okay, or whatever, insert your religion here, okay? Here in Oklahoma and in the United States in general, we get excited because our religious leaders and our parents, they tell us, oh, it tells us with creation and it tells us there's no God and all these things. I'm going to let you know, evolution has nothing to do with creationism. Creationism is a completely separate school of thought. It has nothing to do with creationism, which tells us it has nothing to do with whether or not there's a God or not God or whatever. Okay?
How does it have nothing to do with creationism?
Because all it has to do with is genetic changes over a period of time. Those the ATCG change over time. If we change the nucleotides, we change the what?
So you're talking about
Microevolution,
microevolution
that's
or
what I was
macro?
Microevolution occurs as we go along, and it unfolds into what's called macroevolution, where we can see big changes in it. And so to hopefully better address, evolution is just a change in a real frequency over a period of time. So we're just looking at how these alleles change in a population and over time. And so we're looking at macroevolution over thousands or millions of years. We'll see that genetic evolution, genetic sequences in a particular species has changed. Now we have completely different species. An example would be like a shark. Sharks in the media. You know, they're like, oh, a shark. They call it like a living fossil or something like that, which is poor terminology. Things like that kind of mess with people trying to understand evolution or whatever. But the big thing is evolution is not a religion. It does not have anything to do with creationism. Creation is a separate thing, and obviously there's no solid evidence to support one theory of creationism. But big thing is, I want you to walk out this weekend. Evolution has anything to do with what? Nothing to do with creationism. Nothing to do with religion. It has everything to do with genes and expression in the population. So read chapter 11 over the weekend. We'll cover it on Tuesday, and then we'll get into 12 and 13 next Thursday. Turn in your handouts